Well, October neuroprosthesis seminar. We have a fantastic speaker. He'll get introduced in a second. I just want to alert everybody to upcoming events, relevant events. So November the 5th, the world famous me is going to do the State of the FES Center. Okay? And that'll be a fun event. I'll just say this now before I forget. The holiday FEStival is December 13th. Okay, it's a Saturday, 6 to 10. And it is in the Foundry in the Cleveland Boathouse Bays downtown. Okay, should be a lot of fun, I'm told. And finally, the upcoming neuroprosthesis speakers. Well, we'll hear David Borton in a second. November 13th, Nick Ramsey will be here from Utrecht Brain Center. On December 18th, Rob Franklin will be here from BlackRock Neurotech. And in January the 15th, Jules DeWald from Northwestern School of Medicine will be here. So we have a great set of speakers for you. And with that, I'm going to turn it over to Carl Saab to introduce our speaker.
My pleasure to introduce Professor David Borton, a colleague, but also a dear friend. Could have sworn you've been here in person before, but apparently you gave a virtual talk 2 years ago during COVID, but it felt like so real, I thought you were here. Considering the overlap between what you do and what the FES does, it just made a lot of sense to invite David, and I'm excited to hear his talk today. Bridging the gap. So David obtained his bachelor's in biomedical engineering from WashU in 2006. And then in 2012, he received his PhD working with names that should be familiar to you, Arthur Normico and John Donahue at Brown University. And then he went on to complete his postdoc. It's a Marie Curie postdoc at Lausanne in Switzerland, working with Grégoire Courtine, another name that should be familiar to all of you as well, developing brain-spine interfaces. Currently, Dave is associate professor of biomedical engineering and brain science at Brown, and biomedical engineer at the Providence VA for neurorestoration and neurotechnology. And just recently, in 2024, he became director of the Institute for Biology, Engineering, and Medicine, with the mission to promote interdisciplinary collaborations to face challenges that impact humanity. In short, IBEAM. His research spans from in vitro cell cultures to the design of implantable neurotechnology to translational clinical research, and I've had the pleasure of collaborating with him on multiple grants before. Without further ado, David.
Well, Carl, and thank you so much for that kind introduction. And all of you, thank you for inviting me here. Again, it is the first time I've been to Cleveland in my life, and definitely a pleasure to see all the wonderful work, some of the wonderful work that's going on here. I wish I had more time. I could stay for months to really get to know everyone here and learn all about the amazing work across the clinics, across the multiple centers, and the university, doing really great work in neuroprosthetics. So I've read all your papers, but it's fun to see it all in person. All right, so I'm going to spend, I actually don't know, we have about 45 minutes, something like that, and you'll keep me on time, talking about a particular slice of my laboratory that has been working for the last, let's say heavily in the last 5 years, but maybe the last 10 years on this concept of bridging the gap. And we want to create neurotechnology to replace, restore, and reprogram function across the nervous system. And I'll give you, what I'm going to talk about today is a particular manifestation of that, which is a tool to bridge the literal gap in a spinal cord injury. But the broader technology that we aim to create is bridging a gap between two different areas of the nervous system. It doesn't have to be just across the spinal cord, or, it can be between brain areas or things like that. All right. So without further ado, I'll get started. And as I need to do on every talk, I am here representing myself, only me. So not Intel Corporation, who provided some technologies that I may discuss, not Medtronic, who may provide technologies that I might discuss, and not the VA. I'm not here on VA time. I'm here on Brown University time, and sharing my views as a person, not as a representative of the U.S. government. Further, this work has been funded gratefully by the Defense Department, by DARPA, and by the VA. So I'm grateful to all of their support over the years. Okay, let's get started. My laboratory's mission is to design, develop, and deploy neurotechnology to better understand the nervous system and to improve human lives. That really is the mission that we talk about in the lab, and each student who comes in, we really think about what are we doing that's going to help people. Laboratory, this is at one snapshot in time. These people and the ones I'm going to talk about, the work I'm going to talk about today was mostly done by the ones on the left here, circled in this rectangle. In particular, I'll note that Jonathan Calvert, recently left the lab he was a postdoc and now has a new faculty position at UC Davis working with their neurotechnology, neuroengineering group. And I'm sure you'll hear a lot about his work in the future. And I encourage all of you to take a look at his laboratory as he's starting it up right now. Okay, so this is probably unnecessary background, so I kind of cut out a lot of background slides on the wiring of the nervous system for the biomedical engineers in the room. You're actually all probably neuroengineers, and so you've looked at this in great detail. But I do like to bring up some, inspiring figures in this space. This is Santiago Ramon y Cajal, and as I think about the nervous system and how we are going to interface it, I try to approach it, and I encourage all of us to approach it this way with curiosity and looking at it. He was a neuroanatomist, I guess you'd call him that, an artist, and really just a curious person looking at slides and learning to use these new stains that were coming out at the time to really paint a picture of what these things were in our body that were potentially controlling what we do and think. His workshop looks much more like an artist's workshop, right? And the other one, a sort of engineering companion to that, is Claude Shannon, thinking about information theory, but really another creative person with juggling sticks in his hand, or whatever you call them. He was just a person who really thought about their work from an angle of curiosity and not an angle of like, well, I guess I'm supposed to do this, so I'm just going to do it. So that is how I try to remind myself to approach my work, and I hope all of you continue to do that as well. Okay, so some of these beautiful drawings from Santiago Ramon y Cajal helped us understand sort of this organization of the brain, the neurons, and I guess I should skip through some of this. We have, laminar structures in the cortex, and these laminar structures evolved to big concepts like thinking and motor actions and sensory perceptions. But we also have these structures and motifs in our spinal cord that are doing a lot of work that we don't actually think about all the time, and a lot of, engineering even backgrounds. We don't think about the part down here, and Santiago Ramon y Cajal did actually make these wonderful drawings of spinal circuits, and these are not just, the super highways of conducting information from our brain to our toes. There's processing that happens along this super highway. And we're going to leverage some of this processing to do things like restore motor function. And let's see here. So, yes, it's beautiful structure. I'll continue with that. And much like the internet, we had connections that go from one place to another, one part of the continent to another continent all around the world. And these are one of the first, I think it's the first transatlantic cable was put across this very long ocean, right? And these were just made of copper wires and, horse hairs and some gum trees, I think, saps essentially to coat to insulate the wires. And they lasted for about 2 weeks before they failed. And so slowly we got better at making them. But the bottom line of this is that when this line that connected two very different parts of, let's call it the nervous system, but in this case, the world, when it breaks, people would just go down and they bring it up from the ocean floor and they would fix it and they would put it back down. And we have better technologies to encase it over time. We replace them. So we can think of that like the neurons, I guess. We have these neurons connecting across these continents of our nervous system. And these continents are actually many different continents. And I put this on the side. This is the spinal cord kind of diagrammatically laid out. If I put it vertical, it just gets too small to see anything. But it's a reminder that there's an incredible amount of branching that's occurring as this, we connect from one place to another. And many different connections that are happening, all with millions of sub-connections. And it's a highly complex, highly interconnected system. Okay, an anatomical image of that, which actually we don't all see. It took me a long time in my career to actually look at real human spinal cord. And it's not just that we have these, roots coming out, these big roots that are, kind of obvious to see. If you zoom in under the dura, you have rootlets and all these connections, not just happening at segments of the spinal cord, but like basically all along the cord, these roots coming in. And that's going to come into play as we think about how we're going to interface with this part of the nervous system. And here is the spinal cord sort of cut and put on its side, so we can look at the roots and rootlets coming in from the outside. On the top, this is the dorsal aspect, the sensory fibers obviously coming in there, and the motor fibers coming out the bottom. I think we've all seen this, so I'm kind of skipping over this relatively quickly. So when we have these motifs of circuits in the spinal cord, they're actually useful for us as we learn to coordinate our muscle actions from a child walking around or crawling around, or we're trying to build these internal circuits to respond in a way that is helpful to us to locomote around the world. We have motifs that are going to say, well, if I have if I step on a pin prick or whatever, and it hurts my foot, I want to make an adjustment to my foot position that's going to move my foot out of the way. These circuits are embedded within this gray matter of the spinal cord, and we can activate them through external sensory input, through stepping on something, or we might be able to activate them through electrical modulation, which we'll get into. I'm skipping some slides. It can be, it's actually a much more complicated network, and it has connections that happen across the spinal cord, meaning from left to right. When one foot senses something, it actually is going to impact what the other side needs to do. If I'm going to lift up my right foot because I just stepped on a pin, my left foot should not lift up as well, because then I would fall over. Yeah. So these motifs are built into the spinal cord, and that's really important for us to think about them because they need to be leveraged, or at least taken into account when we interface with the spinal cord with electrical stimulation. Okay. I was trying to get to the point of, okay, we have this wonderful transatlantic cable that we can pick up from the ground and splice back together and put it back. And in spinal cord injury, we have this beautiful organization of the spinal cord, and this wire is going to the brain to do all the different effectors that we're trying to control. But when it breaks, it's a big mess, and it's not something that we yet know how to take up out of the body and splice back together. There are a lot of really interesting, exciting approaches that we could think about in the future of stem cells putting back in there to help reconnect certain areas. But today we don't know what to do with it. And in fact, so spinal cord injury, right? We have lots of different cell types. I should say it's not just neurons that are going to be damaged. We think a lot about the neurons that are in the spinal cord, but there are a lot of other supportive cell types that are also being damaged and put out of place. And it's not so straightforward at how you just kind of would fix that. And this damage that occurs to the spinal cord has an incredibly diverse set of impacts it may have on a person. Here we have your spinal cord injury assessment diagram, sort of so-called ASIA examination, where we might put pinpricks around different parts of the body to say, okay, is this dermatome affected, to figure out what level of injury the person might be at and what function they have remaining. But it's going to be likely a diverse set of problems that these people have after a spinal cord injury. And in fact, it's been studied in many different groups that, when you have an injury like this, it's not just that you need to be able to lift your foot again or walk again. There are many other things that you can't do after that injury. And you have different priorities as what you would want restored to you. So we have, if it's a cervical injury, you'd have, of course, arm-hand function. We have dysreflexia that's happening all the time. You have upper body trunk strength that you want to get returned to you. You want to walk, bladder function, bowel function, sexual function, normal sensations, elimination of chronic neuropathic pain that you might have. So a diverse set of functions. And it's really crazy to think that we can try to envision one tool that would address all of these issues. And I don't know if we really want to necessarily, but it's just a really big challenge that we face from a neurotechnology perspective of how do you create tool or tools to address these very different challenges these people have. It's also an enormous cost to the healthcare system. That's maybe the problem statement. So how is it solved today? Well, today we have, for the most part, the therapy that's provided to someone after a spinal cord injury is to immediately go in and decompress the site to reduce the amount of secondary injuries that you would get from the initial spinal cord injury, and then to put in a rod and screws to stabilize the spinal column so that you're not going to get any more bending or movement around that area. Then you know better than I do, but essentially people go home and they have a rehabilitation routine, which will help maintain muscle strength and existing function or remaining function, but we don't yet have a way to restore function clinically. So, all right, so first question we wanted to ask was, and it's been asked as a field, is can we interact with the circuits below the lesion site? What happens to all those circuits below the injury? So a lot of work has been done in this space to take essentially electrical neuromodulation systems and place them at below the injury site to say, can we reawaken these dormant circuits? And that's really where a lot of, I think, the near-term new rehabilitation technologies will will commercialize and become real viable solutions. But how does it work? Maybe you're familiar with this, but we're taking electrical electrode arrays with, say, 32 contacts on them. And these are placed on the dorsal aspect of the spinal cord. And when we stimulate, we're putting electric fields, charge injection at the dorsal aspect of the column, this is in the epidural space, not the subdural space. So you've got the whole now CSF and the spinal cord's bathed in it. And that's a big conductor that's going to take that field and spread it out a lot. But you can still activate from that stimulation these roots, these dorsal root entry zones, as the maximal tends to be the point of activation where, because of the second spatial derivative of the voltage field, that's going to be the places where you get the highest activation. So as these dorsal roots are coming into the spinal cord, you're hitting them with this spinal cord stimulation, this electric field. So you're activating the sensory system in order to try to activate the essentially reflex arc to stimulate the muscles. So this was it was at an early point done in a rodent model, a double hemisection, as it's called, at different points on the rodent spinal cord. And when you do this double hemisection, this animal has essentially permanent paralysis. There's no regrowth across this double hemisection because it's got to go on both sides. So if you're trying to, is a thoracic, sorry. And if you're trying to have this animal walk, it doesn't respond to a treadmill moving. Its legs are flaccid and does not respond. However, strikingly, if you give this animal a little GABAergic juice there and some electrical stimulation that is tonic stimulation, this animal is able to, the same animal as the last slide, is able to follow not only the treadmill moving just at one speed, but it follows at different speeds. The treadmill changes speed and the animal follows along with it. It has no idea what it's doing. It's completely, from its head, decoupled from the lower limbs. This is just the reflex circuitry being reanimated because it's now boosted up, essentially. So the sensory input coming in from the periphery as the treadmill moves, activating those reflex circuits, and its legs just kind of do what they're supposed to do. Turns out this also works in non-human primates. I'm going to skip a bunch of in-between work here. But if we take a spinal cord stimulator and put it in a non-human primate spinal cord, it's not hemisected, we can enhance their leg movement. Now, what I'm showing here is that in the rodent, you saw that tonic stimulation, meaning continuous stimulation at 30 hertz or 50 hertz, depends, that the animal was able to follow the treadmill. In non-human primates, it doesn't work so well. This is an animal getting tonic stimulation is not hemisected, but the tonic stimulation, as you'll see, is you'll see some competing muscle actions here. It's kind of flexing the limb and it's unable to really extend or flex in a coordinated way. That's what tonic stimulation does. In non-human primates, you need to do a phasic stimulation, which means I'm only going to stimulate at places on the spinal cord that are primarily responsible for the dermatomes, for the musculature of a flexor muscle, for example, during the flexion phase of the gait cycle, and then primarily stimulate a different spot for the extension phase. That's what we call phasic stimulation. So this is phasic of just the flexor, and we're showing that we can enhance the flexion, and we can do the same for just the extension. So an important distinction between what was working in rodents and in primates. The tonic stimulation in primates does not tend to work as well. And now in humans, I don't think I have slides on this, right? Human stimulation, we do tonic stimulation. You can, many groups have shown that tonic stimulation will still boost the spinal reflex system, and you can get the leg moving in sort of like a locomotor pattern. If you get it started, it'll continue for a little bit. But when we're trying to help someone locomote over a terrain, it's really the phasic stimulation that's going to give you that coordinated support. All right, so how did this translate to human work? There was a lot of work done by Cortine's lab and also Reggie Egerton's, Susie Harkema's, and others around what is the right stimulation profile on the human spinal cord. And so a lot of work was to figure out where are these the motor units located on the spinal cord for these different muscle groups that we care about activating. Of course, it was generally known, but to figure out, okay, where do we need then to put the electrodes so that we can activate these different muscle groups at the right time point. So take EMG recordings of a natural gait cycle, which was what we were seeing in the last slide. So a natural gait cycle, healthy subject walking. What are the EMG patterns that we would expect? Or what do we see? Take those EMGs and say, okay, I know those motor neuron pools are here on the spinal cord. So let's now figure out how to put an electrode array that's going to be at those locations so we can activate those hot spots and then activate them at the right time point. So here's this sort of distribution of motor neuron pools and these phasic activation of these different locations. And that enabled this great demonstration that they did back in 2018 in Switzerland of spinal cord stimulation below the lesion site to help someone who was paralyzed locomote with the help of crutches across this terrain. Without the stimulation, this person's unable to walk. And as the stimulation is turned on, again, it's phasic and it's controlled by a Vicon camera, captures motion tracking system to potentiate the muscle that needs to be activated at a particular phase of the gait cycle. So you see this kind of complicated spatial temporal modulation on the left side here. So at the same time it was shown in Switzerland, it was shown by others, including Calvert here, Jonathan Calvert. He was a PhD student at the time at the Mayo Clinic. Oh yeah. So it could also be locomotion not just in the laboratory, but locomotion out in the environment, which was great. So we then started asking the question, could we somehow provide sensory feedback along with this motor activation, or could we use spinal stimulation to provide sensory feedback? And so I'll try to dive into that for a little bit here. And, oh, right, there's a video. Why is sensory information so important when we're thinking about motor control? And here's a little video, but the audio might not work so well. Old video.
He has no sense of touch below the neck, and he's unable to sense where his limbs are or how they're moving. With the help of neurophysiologist Jonathan Cole, he's become the only sufferer from this rare condition to regain his mobility.
Ian has lost this odd thing called proprioception, which is the sensation of movement and position sense. So that if he doesn't look at an arm, he doesn't know where it is.
When you stand and when you dance, you don't necessarily think about where exactly am I in time and space. I have to think about all the processes that go on behind that, developing the movement. Am I safe? Am I in the right position?
And then slowly he realised that if he looked at a moving part and then thought and willed it to move, he could control that movement.
So, as you all know, sensory information is incredibly important for us to help lower our cognitive load in doing the actions we need to do all the time to control some effector of our body. If we lose that sensory input, it becomes incredibly difficult for us to do the actions of movement without wholly focusing on it like he was. So the idea of sensory feedback to a system here is to help someone be able to do their daily activities and reduce the cognitive load of doing their motor actions. It's also, of course, to perform skilled motor actions. The whole separate side of this, if you can't perform a skilled motor action without sensory feedback. But so somatosensory feedback is critical for motor control. And this person, right, the need for concentration was total. He cannot emphasize enough the effort needed to build up in his mind every move and counter move to kind of get through his day. And of course, there are many more falls of people who have lost the ability to sense. So we could put in sensory information at a lot of different locations. You guys here have studied this in many different ways. You do, I believe, intracortical microstimulation for sensory restoration now, and that's really great. You could put it in through the spinal cord, which I'll talk about, and you could put it in through the peripheral nerve, which you also do quite a bit of here. So I'll talk about epidural stimulation on the spinal cord to provide sensory feedback to people with spinal cord injury. So, it's kind of interesting, right? We were already using spinal cord stimulation to do motor control, but it was through the sensory system there. So we should be thinking now, how can we do sensory stimulation or spinal cord stimulation to provide sensory feedback? It's a natural place. You're already hitting the dorsal column or the dorsal roots going into the column, so it should likely provide some sensory activation. Let's see here. Yeah, what happens to these percepts, the potential percepts, if we're stimulating the spinal cord? We're going to hit these dorsal roots, but those are, of course, going to go up through many different structures as they finally get to the place in our brain that's going to help us make the decision, or a participant making the decision of, like, I felt something. And it's important to remember that the sensory process, while bottom up in that the sensation comes from the outside, it's also top down, and that we're having to interpret what that sensation is as a person, the mind part of this. And did I feel it? How much did I feel? It's not, there's the bottom up and the top down part of that. Okay, so we started by doing some work about how do we just even look at the translation or transformation of spinal cord stimulation to sensory cortex dynamics. We want to understand, if we're going to stimulate on the spinal cord for sensory restoration, what's happening in sensory cortex, and is that somehow related to the perception that someone or something is reporting? So we had a study that was done a little bit ago. It's still sort of in process of being written up, where we had non-human primates with a spinal cord stimulation system in them that Medtronic kindly collaborated with us on, and a brain recording system that we could put in a Utah array. This is a bed of 100 needles, 96 needles that go into the cortex, in this case sensory cortex, to then record while we were stimulating on the spinal cord. So we get lots of LFPs, single units, all sorts of stuff. And leg movements are represented in sensory cortex. Of course, we're in leg area of sensory cortex. We hope that there will be leg area activation. But the question is really more about, how does the stimulation affect that existing dynamics of that population of neurons? So here's the system we put in. This is the Medtronic, it was called the RC+S at the time, and an electrode array that we custom designed to fit, again, to these kind of mapping that you would imagine, just what I showed you on the humans, to map onto the non-human primate lumbosacral region of the spinal cord for lower limb. So the question there was, well, does SCS modulate these patterns of activity in the brain? And if so, then how does it modulate it? There's kind of two ideas here. One is that if we stimulate the spinal cord, that our sensory cortex dynamics, which are representing the leg movements, that the stimulation is going to just, think about dynamical systems, it's going to push it further along some existing axis of that sensory cortex activity. Or if we stimulate, is it going to take this these dynamics, or this point of the space the current thought is, into an orthogonal direction, where the animal would not be interpreting it as a sensory percept, but instead as a paresthesia. And so that was sort of our question. I won't go into all this. Did a lot of recordings, a lot of stimulations. Our essential point of all of this, or just distillation of all that work, was that here's a chart here. It's about cross correlations. Don't worry about that. Let's get some distance measures here between states of the brain under different conditions. These are dynamical states. So we have a sort of baseline condition where the brain is just kind of hanging out. We have a M state that's read as the movement state. So when the leg was being moved, that's the kind of the natural state. And then we have a stimulation state, which was over here is baseline. Stimulation state is down here in blue. So if we stimulate and do not move the leg, the brain's dynamics go down here. And if we stimulate while the leg is moving, the brain dynamics go up here. So these are unitless distances here. So what's more important is that these are in distinct clusters, and that our stimulation profiles that we had time to evaluate in this study, which were fewer than we would have liked. It was a complex study, getting spinal cord stimulators and brain recordings and all this stuff. But in that evaluation, the spinal cord stimulation is not yet in line with that manifold that the sensory cortex is on in a natural movement. And that makes sense in a way, because when we stimulate in humans, that I'll get to in a minute, generally that stimulation is interpreted as a paresthesia, a sort of tingling or non-supernatural type of sensation. I would love to find in the future some way for us to, in a principled way, find approaches to stimulation of the spinal cord that get us to the natural state so that we can then predict that this stimulation profile will give a natural sensation to a person. But we're not quite there yet. I think actually in talking to Balu today, it was nice. You guys have recordings in sensory cortex in human participants and peripheral nerve stimulation, which you could sort of get at some of these questions. It'd be really fun. Okay, so we then, after the monkey study, we then decided to run a study in human participants, where we're stimulating now, instead of below the lesion site, stimulating above the lesion site to provide a sensory feedback to them and to really quantify what sensory perceptions they get from these different stimulation inputs. So there were 2 participants in this part of the study. I'll label them as P2 and P3 for reasons that will be clear later. But and these participants had, the first one here was complete spinal cord injury, 52-year-old male, Asia A, 10 years post-injury. Participant 3 was Asia B, T5, so incomplete, sensory incomplete. And we put electrode arrays up at this boundary between the cervical and the thoracic. The actual location really depended on the injury and the neurosurgeon's comfortability of getting close to the injury site. Yes. So in this study, we were very curious about, what these perceptions felt like. So we wanted to actually give a lot of the control of the stimulation over to the participants of the study. So we developed a DJ type interface that allowed the participants to manipulate unknown variables to them, but here on the table, participants are manipulating the frequency and amplitude in these 2 axes, and on the right side is the electrode number. So they're kind of playing this game and searching this big sensory space, and each time that they feel a different sensation, something changed, they go over to this percept mapper, it was called, and Pitt developed it, and we kind of adapted it from their software package to have the person label where I felt what and what it felt like to quantify the feeling. So we did both a table-based DJ and then we built a tablet-based DJ for this other participant later on. And here's that percept mapper. It's like, how do we quantify how something feels? And it's a lot of great work that was done by Pitt to develop that. And then what we did is took this, extended to the lower limbs, which is not shown here, but also made a mapping of the stimulation points onto dermatomes and then put that into a sort of file type that we could ingest into a neural network that I'll get into in a minute to help train some models. So what percepts do we get? So when we stimulated, this is again stimulating in T1-ish area, obviously we're getting upper thoracic. Now this person had a T5 injury and we're providing sensory feedback in the upper limbs, right? That's a little disconnected. We're not providing, and the goal wasn't ever to provide sensory feedback of the lower limbs. Like they're bound accord with their complete injury. They're not going to have a lot of fibers going back up to their brain from those spots. However, the goal would be to replace a sensation, like you just saw that person walking without proprioception. If he had anything that he could then relate to his foot position, his ankle angle, knee angle, things like that, that would likely lower his cognitive load. And that would be the kind of mission of this technology as it moves forward. In any case, we get a relatively diverse set of activations across these dermatomes, a set of different qualitative responses to it, some being natural, some being sharp, tapping, electric flutters, tingles, pulses, vibrations, and things like that. Let's see. We then asked them to do a cognitive load task, but I don't think I'm going to talk about that. Oh, so hold that extra to the side. We asked them to, in this control of the stimulation that they were doing, we wanted to derive from that the discrimination thresholds for them. And so as part of, they played sort of a slightly different game that was a little more tuned for us to figure out where their detection thresholds were at. And looking at, well, how does pulse width affect that? How does amplitude affect it? How does frequency and those sorts of things? And suffice to say, it's kind of diverse across participants. This first one, now they're labeled as P1, P2. Apologies for that. This is P2 as I defined it before, and that's P3 as I defined it before, but it doesn't matter. 2 participants here. And so it's diverse. So in P1, who was the 52-year-old who had 10 years post-injury, he was also on a lot of baclofen. His detection thresholds were very high and somewhat unclear. Like it's just like very wide uncertainty about when he was going to detect something. Whereas our P2 was highly focused at this, 360 sort of amps pulse width, nanocoulombs being injected. And that's interesting. So I think there is going to be an inherent diversity based on the injury, based on the time of injury, based on the person's drug regimens and all that in these detection thresholds. Of course, the individual threshold is going to be different, but like even the statistics around that threshold are going to be quite different. And that's going to be interesting to deal with. The other takeaway from this that I'll share now is that long duration and short duration stimulations are very different in how they get perceived by someone. They're even here. A long duration stimulation was starting to activate things that wrap around the back of their back, whereas short duration was just in the front of their chest. And it kind of makes sense. You're slowly recruiting more fibers. But it's something we need to take into account if we're building a sensory prosthetic that is going to be dynamic to the environment. It's going to be tricky to figure out what is the right stimulation to represent a salient sensation for that person. And that it's going to change over time. If that stimulation stays on for a certain amount of seconds, which is normal for a sensory process, that sensation may change. And so we need to somehow account for that. One way we've been thinking about accounting for that is developing biologically inspired neural networks. And so we partnered as part of a DARPA program with Intel and with some collaborators at Brown who are experts in building computational models to think of a way to infer stimulation parameters that will be stable to achieve a particular target outcome. That target outcome could be, in this case, I'll describe it as a sensory percept and a stable sensory percept over time. So what do we do with that? I guess I'll step back a bit and say, if we build a black box model, a neural network model to do this, you need a lot of samples for training data. You have a lot of weights you got to train for or optimize around, and that's going to take you too long, in our view, in a clinical study. Like you just don't have time to collect so many samples. And so we thought we needed to take an approach that was a little more biologically informed, essentially constrained to the architecture. Well, the question would then be what to constrain it to. So we said, okay, let's constrain it to the architecture of the spinal cord. So we spent a long time thinking about how the spinal cord is organized and trying to derive some central motifs that we could build into our neural network model with a bunch of gated recurrent units and things like that. A model that we called Deep Spine. And the goal of it, though, was to just essentially reduce the number of weights that we have to train, which then reduces the training data we need to get it to perform. There's some other ideas that maybe would be more resilient to outliers that might come from the training data sets. So we built this model first in sheep, using sheep. We were doing a similar experiment, of course, not with sensory percepts in sheep. That'd be a little complicated. This was with motor activation as a target. And I'll get back to motor activation in a little bit. But just imagine they were sensory targets. It doesn't matter the target from the neural network model perspective. The targets here were EMG activity. And what we do is we build a forward model first to say, okay, we do stimulation and we get an EMG output. We build a model, we train our neural network on those associations. And then we build a second model that goes backward, the inverse model, to tell us how can we get stimulation parameters that we want in order to get a particular target EMG. And that's really helpful because that inverse model gives us this big space, this probability map here of where am I likely to get the target output? What stimulation set of parameters will give me the highest likelihood of achieving a particular EMG output? What's really nice about that is that this particular one's not a great example. This is like, everything kind of comes up to one bump in the middle here of some parameter space, but it's not always like that. So I should have brought in other examples, but some of them have 2 bumps. Some of them have like odd shapes in this probability space. And that's important because I can then choose a parameter set that's going to have an equal potential of getting my target output. And I can choose that particular spot in the parameter space that minimizes my side effects, for example. And that would be really hard to do in a principled way without having some model like this to help infer these parameters. It's also really helpful because when we're getting a system like this out in the wild and we're trying to retune over time based on new data that comes in, we now have a model to update weights on. And that's going to be helpful. Okay, so we did this model building for our sensory activation in humans. So we built a forward model, built an inverse model, and then asked the system, we want to activate this part of the body and as a sensory percept, not as an EMG now. And here's this inferred parameter space, and we can choose our peak there if we want, whatever. And then we can use those parameters and ask the person, what did you feel, right? And so we do that, and we get high correlations with that. The best real way to show that, though, is to have a person use that percept. And in this case, we ask the participant, which percept do you want to use, right? This is the whole kind of cycle of this. It starts with the participant, right? What percept do you want to use to represent your joint angles here of your knee, for example? So they were asked to use their hands to report out the knee angle that we're moving the knee at. And they can't feel anything below there. It's blindfolded, all that stuff. So they got to choose the percept they wanted. It could have been any percept. They could, well, within reason here, but under the arm, on top of the arm, on the chest, whatever. And then the model will say, okay, that's the percept you want. Here are the parameters. We put those parameters into our system that's doing this closed loop recording and stimulation. And here they're using that for now bilateral. So she chose 2 percepts, one on each side to use, and she can report very high accuracy, which limb is being moved and what angle the knee is at. It's just an intensity of some sensation, right? It's a sensory replacement study. But it's done in a way that allows us to envision how this would work at home and how it would be trained over time because of this model-based approach. Okay, now the next goal we had was to think about how could we do, or should we do, paralesional neuromodulation. Should we stimulate to get motor activation, or can we even stimulate to get motor activation below the lesion site at the same time as providing a sensory feedback? So that's where this bridging the gap sort of comes in a little bit. We now have a tool that is spanning the injury site where we can provide the sensory feedback above the lesion and provide the motor activation below the lesion. In the future, we'd like to see this system be fully implanted and all these fancy neural network models running on device and all that sort of thing. Right now, in the study I'll show you, this was all percutaneous in a 2-week inpatient study. So patients came in. They had, the ones I think I'll show are 2 complete injury subjects. They had paddles put in the T1 to T3 and paddles put in T11 to L1. Yeah, I'll focus mostly on participants 1 and 2 here, but there's 3 participants in the full study, and here are the kind of x-rays. You can't maybe see the injuries so clearly. That's unfortunate, but suffice to say the injuries were anatomically complete in these subjects, not just ASIA complete, which is distinct. So we first, I'm not going to go too far into this, but we do recruitment curves of the lower limb activation, so the stimulation from below the lesion electrodes. You've seen it probably in many papers already. You don't need to go into that, but you stimulate at different electrodes and you figure out what muscles were activated. And we then use the same methodology of neural network model optimization. So we built a lower limb stimulation forward model and then built an inverse model to infer the parameters we should use to activate particular musculature of the lower limb. So we then demonstrated the lower limb stimulation control side of it by having the person now move their arm to control the legs, kind of the opposite of what we showed in the sensory side. So he's just moving his arm up and the leg will flex. He's going to move his arm down and the leg will extend. He had spasticity. You'll see some shaking of his limb. He has spasticity all the time. That's sort of his, yeah. But so we can do this. We can do it with both hands, moving their legs up and down. And that demonstrated is all based on the neural network parameters that were chosen. So if we want to activate these limbs, it infers the parameters, and we use those parameters on the computer. Okay. Okay, right. We're showing that here it was very accurate. The timing of the stimulation, activated the correct muscle groups to move the limb, which is kind of seen by him actually moving the limb. So then we wanted to show somehow that we could do these at the same time. It's not clear that that it would work in a way that they would be able to perceive the sensations in the same way when we stimulate above the lesion when they're in the middle of stimulation below the lesion to support their locomotion. This is somewhat of a contrived setup, I will say, because the person is a complete injury. They have no way to get trunk support from this or body support. So they're in a body weight support system, and their legs are being moved by a rehabilitation specialist, two of them, to just help the legs be in a locomotor-like state. The readouts of this are essentially that we see EMG activation of the proper muscles, and that the rehabilitation specialist will report that they had to do less work in their movement, right? But that's very qualitative. It's unfortunate in this sort of setup. But he's going to be reporting, when his foot is touching the ground on the left side and the right side. Now, it's clear to me that he would be able to do that even if we didn't stimulate, because there's, the foot's touching the ground. There's a lot of vibrations through the body, and he has feeling above the chest. But we ask them, right? They're using the stimulation as cues of when their leg was touching the ground and which leg it was. And we have had them also report how bent the leg was at different time points during this. It's hard for them to do that with their hands in this setup because they're having to support their locomotion. But the crux of that story is that we could perform meaningful sensory feedback. They could use that sensory feedback to report which leg was being touched on the ground during meaningful motor activation, activating the right muscle groups for locomotion, flexion muscles and extension muscles. So we also asked the question of, well, could we be using this stimulation to activate the autonomic system, and in particular to help rescue blood pressure changes that may be occurring in these participants. So what I'm showing here is a participant. This is a healthy participant doing a Valsalva maneuver. So that's when you're pushing hard on your belly. And what you normally see in this procedure is that the blood pressure drops, but you see a commensurate increase in the heart rate, and you're going to, rescue your blood pressure over time. The vasoconstriction is going to occur, and you're going to, heart rate's going up, blood pressure goes back up, and you're able to maintain a stable blood pressure in your body. In spinal cord injury, that's what that says here. Yeah. So here the pressure drops, but not that much. There's an increase in heart rate. We're not showing ECG on that one, but it's you can kind of see it reflected in the blood pressure because blood pressure is high enough. When you have a injured participant, it was P2 here, when they do a Valsalva maneuver, their blood pressure drops, and there is not an increase in the ECG. And if you kept them doing the Valsalva maneuver, they would eventually pass out here. So we only do it for a certain amount of time. When we give spinal cord stimulation to a particular location, what we see is we have the blood pressure drop, but it's maintained at some level that we still are able to see these pulses coming in from the heart rate. What we do not see is a heart rate increase. And that is because we think that we are not really activating the autonomic nervous system in a way that would be natural for the person. We're probably activating musculature, is constricting the blood flow and thus raising the blood pressure. And so this is still, mechanisms are being teased out. There are recent papers by Cortine's lab looking into this mechanistically in rodents and a little bit in humans. I think it's still trying to figure out how is spinal cord stimulation interacting with the autonomic nervous system here. I don't know if we totally know the answer to that. But suffice to say, it can produce quite palpable demonstrations of raising the blood pressure of someone who would otherwise have passed out in this experiment. So that's good. And there's also the possibility that these types of systems in spinal cord neuromodulation epidurally can provide bladder control. That would be wonderful. I know many of you here work on some of this work. We have really only done very surface level measurements here and doing urodynamics in patients of our study and looked at stimulation at certain spots on the spinal cord that will provide detrusor pressure increase and not other locations on the spinal cord. So you have to find these certain hot spots that will be over roots that are going out to the detrusor muscles. But we can find spots that do activate the detrusor. So that's great. This has, we're not getting full voiding. We're not getting anything that you'd really want if you were trying to build a real prosthetic for bladder control and full micturition. But it means that we can activate some of those circuits, and there should be a lot more. We should do some studies specifically on the bladder activation part of spinal cord neuromodulation. Okay, so the next steps of what we're trying to build here is, can we bring this all into a fully implanted system and have a study that lasts for more than 2 weeks? A huge limitation of our studies so far have been their inpatient population. Patients come in and they stay at our hospital. We have a great setup. It's not quite as nice as the one here at APT, but it's a nice setup, or the FES Center. But it's a nice setup. And we want to be able to take our technologies to the patient's home. And for that, in our view, we need an implanted system to do that. And so we are currently developing implanted systems for that use. This is one version of that has been evaluated in large animals for extended periods of time, years. And this is a combination of a lot of different technologies that well, we have 9 minutes left. I could talk about some of them that I think are relevant to what you look at here. So you have this network neuroprosthetic system that has been homegrown in this area by Hunter Peckham and the team, and you have the system by Dustin Tyler and his team looking at another way to connect multiple systems in the body. And this is, I guess, of a similar concept. Our view is that we need to have high flexibility in an implanted system to be able to meet the needs of researchers and actually, I think, clinical targets. So this system, this device has high capabilities, meaning high data rate transmission, hundreds of megabits of data that it can send out, and it has programmable logic on the inside to run neural network models that we've programmed. We had worked with Intel for a while on building customized silicon to run efficient models for this application. Sadly, Intel's a company, and they're not listening. It's okay. I'm not talking for them. They have companies change their directions often. They've changed leadership. They've basically almost now the U.S. owns 10% stake in them, whatever. But nonetheless, I think there is still opportunity to build specific neural network engines. Let's call them that, tailored to these purposes, because we're going to need to use them to do a lot of the exciting new decoding and I think stimulation paradigms that we want to do. So this has that type of technology on it. It also has the ability to connect to active electronics outside of that device. So these leads here are actually 64-channel leads that have hermetically sealed multiplexers on them that allow us to connect to 64 channels more or less at one time, sampled, of course, multiplexed, to both record high-fidelity signals from these electrode arrays, but also to stimulate at many different arrays at different times, many different electrodes at different times. This technology is FDA approved at the moment, meaning we have an IDE, FDA approved IDE, for putting those in human subjects, and we're very excited to take that next step to have a first active electronic on the spinal cord, which would be great. And this system also has a fabric for connecting different devices. It doesn't have to be this one electrode array. If you reprogram it, it can connect to many different peripherals, much like the, I think it's the networked neuroprosthetic system that's been built up here. So I think this paradigm, it sounds like a lot of different groups have over time continuing to build momentum around a interconnected system of systems in our bodies that are going to help us solve these more complex challenges like treating people with spinal cord injury. Yeah, I think that's all I want to do there. Okay, so what we want to create is this single unit device that can adapt to the needs of the nervous system. We could be using it in the spinal cord only to bridge a gap in spinal cord injury. You could be using it in the brain to record from the brain and stimulate the spinal cord, and to maybe just be in the brain, 2 different areas of the brain to help with stroke rehabilitation or other mental illnesses and things like that. But I think building in the intelligence piece, the fabric for doing complex machine learning is going to be important. And that's not something companies really want to do. So it's something academia is going to have to push a little bit on. All right. So with that, I'll thank the people who have really done all this work, and in particular, the participants who gave their time. They got nothing out of this, right? They came in for 2 weeks, actually much more than that in terms of all the preparatory work and post-op stuff, and really gave us all their time and energy. So I want to thank you all for listening. It's been a pleasure to be here, and I'm really happy to entertain whatever questions you have about this work. Thank you.
Questions for Dr. Borton? Yeah, to me.
55, yeah, good.
Yeah. I forgot. We have online people. That's why we have to have.
Oh yeah. Okay. Yeah. Oh, was this online? I didn't share Zoom or anything. I guess it's through your system. Okay. Good.
Thank you for the talk. Specifically about the sensory restoration, sensory stimulation. So you're using a sensory substitution, right? Substitute proprioception using cutaneous sensation above the lesion. With the work that you have done with the subjects, is there a sweet spot that they preferred, for example, shoulder versus arm versus chest, that they felt, oh, this really works well with the leg movement? And also in the video that you showed that the person was walking and they were reporting their leg movement, did they find it useful, or was it an extra thing that they had to think about?
Yeah, great questions. So we did, I don't think it's published yet, but there's a paper that we have about the qualitative aspects of the study, where we had a lot of interviews with the participants and really asked these types of questions of how useful things were for them. How would they envision using it in their daily life and things like that. And so all the participants were very clear that this, while it may feel different, like it's my chest being activated, but I'm supposed to associate it with my foot, it feels different at first. They all uniformly thought it would be they would integrate that. It felt more natural over time to them. Like they could, it felt more like it was just their leg moving even within the 2 weeks of the study. And they felt that would just continue post-study. Who knows what that would bear, out, but that's, I think, why we want to do a long-term take-home study of this type sort of thing. It's a really interesting question about the preferred sensation. We just didn't have enough time. We had 2 weeks where we were doing a bunch of different experiments, and we did not have time to explore that question of giving them, a whole day or half a day to explore which sensations they preferred for this movement. So I can't say that they had a preferred spot. That's a good question, though. Yeah.
Fantastic, Dave. Oh, that's fun. My question has to do with the motor stimulation in the spinal cord. So you're going through the sensory system, and in the deep past, people stimulated the peroneal nerve to evoke flexion withdrawal, which is similar, I think, to what you guys are evoking. But accommodates. I mean, reflexes accommodate. They have it over time. Do you have that problem in the?
So in this, what I've talked about today has been short-term stimulations. So I don't think we saw.
Hold on. I mean, like over minutes.
Yeah, well, we're only stimulating, short stimulation and only very infrequently. So even over minutes, we're only stimulating, a couple of times. We did not. So maybe to answer your question, we did not see changes in the activation, the stimulation necessary to activate the muscle, but we did not look at the amount of activation over time of each muscle. So we could look at that and see if we're seeing a small amount of accommodation. Yeah, that was not. We didn't look at that. So we have the data, so we can definitely look at that. Yeah.
So you said that you noticed two different locations on sort of the pulse frequency, pulse amplitude space that would provide the same, ideally provide sort of a confident level of EMG, right? Not shown in that plot, but that there were times when you saw that.
Yes.
So at least in peripheral stimulation, it's generally accepted that increasing either pulse frequency or pulse amplitude is going to increase the overall activation, the muscle activation, pretty much monotonically. Mm-hmm. Do you have an idea, is it because we're in the spinal cord that that's happening, or do you have an idea what the mechanism would be where it wouldn't be monotonic increases, but there would be two sort of nodes?
Yeah, I think it's really about the spread of current in the cord, the field there. It's you got this whole cord, the dorsal roots are pretty far away from your stimulation, and so you got a lot of spread that's not going to be as linear. You on the peripheral nerve, you're like your cuff's right on it, and you're just getting a clear activation of that one nerve. I think in the spinal cord, it's much more diffuse right now with epidural stimulation, and I think that's both a benefit and a drawback. You get these opportunities of maybe activating the same circuitry with different side effects, but you have less ability to specifically activate one thing, one muscle group.
Thanks.
Thanks, David. I have one question that's a clarification, and then maybe one that you could speculate on. It might be related to Ben's question, but when you were showing your participant moving their arm to drive the motor stimulation for the leg, that was position, right? We're using the Vicon position, so that could be any input. It could be a dial, it could be a button switch, it could be, okay, so it's not the fact that it was motion to motion was for demonstration purposes, right?
Correct.
Okay.
I think it is different from
an EMG, it was the actual elbow angle.
Yes,
right?
it was elbow angle. I think there is a distinction, though, when a participant is controlling it versus us as a researcher. I could have had a dial there and it's gone like this, right?
Yeah.
I think that it's different from a participant's perspective. And I think in the end, we do want to get to places where we're having participant-driven neuromodulation.
Right.
Yeah.
And I guess my question for you to speculate on is, I think you alluded to some of this, but I often wonder what happened to intraspinal microstimulation. You're on the epidural, in the epidurum, and there was a lot of activity a while ago where people actually penetrating the core to get more precise locations. Is that, what's the advantage of this approach besides the surgical implementation, which is routine now, right?
Yeah.
How do you see the sort of intraspinal micro stimulation playing out? Does it have a role? What advantages did you have with this epidural approach over that?
Yeah, great question. And I have some thoughts on it. So epidural stimulation, like you said, is surgically very easy. In a broad sense, and is accepted by the risks are well known. I guess put it that way. The risks of intraspinal, just putting something intraspinal, are not well characterized. Like there's a lot of different things that could go wrong, but that shouldn't stop us from exploring it. But the benefits there are that you're going to get much more specific activation, and you can even go to the motor neuron pools directly if you want. You could hit sensory specifically. You could do a lot more tuning, which would be really exciting. The tools available to do intraspinal are not great right now. Neuralink is exploring it. There'll be threads put into spines. Maybe they already have. I think there's an interesting medium that hasn't been as well explored either, which is the subdural, getting at the rootlets themselves, but not in the epidural space, but putting something down. Now, to do that, you have to get, you don't want a cable going through your dura the whole time. That's too dangerous. So you need a wireless system subdural. And there have been some groups that have looked at that. I have some ideas on that. I think there's an opportunity there to get much better specificity and without the complexities, without the risks.
One more thing. I guess you went through it pretty quickly, but when you saw the autonomic changes in blood pressure, where were your paddles? Were they in the cervical region or high thoracic region? What was it, the same region you were getting the percepts from, the sensory percepts?
Right. They were in. No. They were stimulating T. They were below. They were in the caudal.
Below the injury.
Yeah. Mm-hmm.
Okay.
Mm-hmm. I can't remember exactly which electrode and root, but it was T12. So where they're at. Yeah. Mm-hmm.
Questions?
We are right on time, everyone. This is great.
Thank you.